Hello and welcome back to Suchitra's Biology. Hope you all are doing good. So today we are going to discuss about the topic that is glycolysis. So let's discuss it. Glycolysis. It is a topic under the biochemistry. Okay. So glycolysis occurs in cytosol in both prokaryotes and eukaryotes. Okay. In both prokaryotes and eukaryotes, glycolysis takes place in the cytosol. Okay. Now, what is glycolysis? Glycolysis is a 10-step reaction. Okay, 10 steps. There are 10 steps in the glycolysis. Now, glycolysis can be written as glucose converted to 2 molecules of pyruvate plus 2 NADH plus 2 net ATP. Actually 4 ATP are produced, but as 2 ATPs are also used, so the net production of ATP is 2. Okay, so this is the overall reaction of the glycolysis. Glucose is converted to 2 molecules of pyruvate, 2 NADH and 2 net gain of ATP. So let's discuss the glycolysis in detail. As I said, it has 10 steps. 10 steps, okay. 5 steps and 5 steps. Okay. In these first 5 steps, energy is given. Energy is utilized, you can say. Energy is utilized, and in the last 5 steps, this is the payoff phase. Payoff phase, during which ATP or energy is produced. Energy is produced. That is, ATPs are produced in the first 5 steps. The ATP are utilized. In the next 5 steps, the energy or the ATP is produced. Okay, this is the overview. This pathway starts with glucose, right? So glucose converts to glucose 6-phosphate. Glucose 6 converts to fructose 6-phosphate. Fructose 6-phosphate converts to fructose 1,6 bisphosphate. I have written this P as it is phosphate actually, okay. I'm not writing it completely. So glucose, then glucose 6-phosphate, it gets converted into fructose 6-phosphate, then fructose 1,6 bisphosphate, then glyceraldehyde 3-phosphate and dihydroxy acetone 3-phosphate. Okay. Glucose is a 6-carbon molecule. Glucose 6-phosphate is also 6-carbon molecule. Fructose 6-phosphate is also a 6-carbon molecule. Fructose 1,6 bisphosphate is also a 6-carbon molecule. Now, the fructose 1,6 bisphosphate is cleaved into 2 molecules. Okay. Glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. Glyceraldehyde 3-phosphate is a 3-carbon molecule, and dihydroxyacetone 3-phosphate is also a 3-carbon molecule. Okay. This was 6 carbon. It gets split into 2 molecules, each of 3 carbon. Now, the glyceraldehyde 3-phosphate gets converted into 1,3 1,3-bisphosphoglycerate. Okay, this gets converted into 1,3-bisphosphoglycerate. Then this gets converted to 3-phosphoglycerate. 3-phosphoglycerate. And the next step I will write afterwards. Okay, so this is the first step, this is the second, the third, this is the fourth, this is the fifth, this is the sixth, this is the seventh. As I told, the first 5 steps are energy utilization steps. Okay, these first 5 steps. Okay, the next 5 steps are payoff phase. So glucose is converted to glucose 6-phosphate by an enzyme known as hexokinase. Hexokinase. What is kinase? Kinase is the enzyme that adds phosphate, right? It takes phosphate group from ATP and it adds to the molecules. So hexokinase takes phosphate from ATP, right? So 1 ATP is utilized here. Now glucose 6-phosphate is converted to fructose 6-phosphate by enzyme Isomerase is what? It converts one form into the other isomeric form. Glucose 6-phosphate and fructose 6-phosphate are isomers, right? So this is isomerase. Now fructose 6-phosphate is converted to fructose 1,6-bisphosphate by an enzyme PFK1. It is a very important enzyme. PFK1 stands for phosphofructokinase. Again, it's a kinase. So it adds phosphate group, right? Fructose 6-phosphate has only one phosphate group, but fructose 1,6-bisphosphate had 2 phosphate groups. So PFK1 adds one more phosphate to the fructose 6-phosphate. It again consumes an ATP. Okay. Now the fructose 1,6-bisphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. Okay, so I was explaining that this fructose 1,6-bisphosphate gets converted into glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. These two are 3-carbon molecules, right? So fructose 1,6-bisphosphate gets converted into glyceraldehyde and dihydroxyacetone by an enzyme known as aldolase. Aldolase are the enzymes that cleaves some complex molecules into simpler ones. Okay. Now this glyceraldehyde 3-phosphate and dihydroxyacetone are interconvertible and are catalyzed by the enzyme known as triose isomerase. These two are also isomers, so it is catalyzed by enzyme isomerase. Triose phosphate isomerase. Now the glyceraldehyde 3-phosphates gets converted into 1,3-bisphosphoglycerate. Okay, so these 5 steps are energy utilization steps. Okay, the next 5 steps are payoff phase, or where the ATP or the energy is produced. Now glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate. It is catalyzed by an enzyme glyceraldehyde 3-phosphate dehydrogenase. Okay, glyceraldehyde 3-phosphate dehydrogenase. Okay, as it is a dehydrogenase, it is involved in the production of NADH. 2 NADH from 2 NAD plus. Okay, so 2 NADH are produced in this step. Now the next step is 1,3-bisphosphoglycerate converts to 3-phosphoglycerate. In this step 2 ATPs will be produced. 2 ATP produced from 2 ADP. Okay, from 2 ADP 2 ATPs are produced. As I said, these 5 are the payoff phase. So energy is produced. Now the 3-phosphoglycerate gets converted into I will first erase it and then we will see. Okay, three phosphoglycerate 3-phosphoglycerate gets converted into 2-phosphoglycerate 2-phosphoglycerate converts into phosphoenol pyruvate, and phosphoenol pyruvate gets finally converted into 2 molecules of pyruvate. Okay, three phosphoglycerate gets converted into 2-phosphoglycerate, then 2-phosphoglycerate gets converted to phosphoenol pyruvate, and phosphoenol pyruvate gets converted to pyruvate. Okay, 3-phosphoglycerate is converted to 2-phosphoglycerate. It is catalyzed by the enzyme mutase. Okay, this is the 8th step, this is the 9th step, and it is the 10th step or the last step. Okay, so mutases are the enzymes that remove the phosphate group from one position and add the phosphate group to another position. Earlier the phosphate group was present at 3rd position, but mutase does what? It attaches the phosphate group to 2nd position. Okay, so it just changes the position of the phosphate group. Now the 2-phosphoglycerate gets converted into phosphoenol pyruvate by the enzyme enolase, and here 2 molecules of H2O are released. Here also 2 ATP are produced. 2 ATPs are produced from 2 ADP. Okay, and then phosphoenol pyruvate is finally converted into 2 molecules of pyruvate. Okay, so this is the glycolysis pathway. Now let's see the summary of the glycolysis. We have seen 2 ATPs were used, 2 ATP used in energy utilization phase, and 4 ATPs produced in payoff phase, right? So the net gain of ATP is what? 4 ATPs are produced but 2 ATPs are used. So net gain of ATP is 2, right? 4 minus 2 is 2. If only asked how many ATPs are produced, then you have to answer 4. But if it is asked what is the net gain of ATPs in glycolysis, so the answer will be 2. Okay. These 2 ATPs are used in the first step and the third step. First step and the third step. Okay. Then 4 ATPs are produced. 2 ATP in 7th step and 2 ATP in 10th step. Okay, I have done a mistake in the earlier slide. Okay, that I have mentioned the 2 ATPs in any other step, I guess 8th step, but 2 ATPs will be produced in the 10th step and 2 ATPs will be produced in the 7th step. Okay, so this is the 2 plus 2, 4 ATPs produced and 2 ATPs used in first and third step, so net gain is 2 ATP. Also 2 NADH are also produced. 2 NADH is also produced. So glucose converted to 2 molecules of pyruvate in 10th step plus 2 NADH plus net ATP plus net gain of 2 ATP. Okay, and water is also released, 2H2O. This is the overall reaction. Okay, I will explain it once again in a simpler form. Glucose 6-phosphate, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-phosphate, fructose 1,6-bis-phosphate, then glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate and dihydroxy acetone 3-phosphate. Then glyceraldehyde 3-phosphate was converted to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate was converted to 3-phosphoglycerate. 3-phosphoglycerate was converted to 2-phosphoglycerate. 2-phosphoglycerate. 2-phosphoglycerate was converted into pyruvate. Phosphoenol pyruvate. Phosphoenol pyruvate. Then phosphoenol pyruvate was finally converted into 2 molecules of pyruvate. Okay, so this was the reaction. 1, 2, 3, 4, 5, then 6, then 7, 9, and then 10. The first step, second, third, fourth, fifth, sixth, seventh, 8th, 9th, and 10th steps. Okay, glucose was converted into glucose 6-phosphate and 1 ATP was used, right? 1 ATP was used. Then also remember the first step, the third step, and the 10th step are irreversible in nature. Rest all steps are reversible. Okay, which steps I said? First step, third step, and 10th step are irreversible. Rest all steps are reversible in nature. Okay, now glucose converted to glucose 6-phosphate by using 1 ATP. Then glucose 6-phosphate converted to fructose 6-phosphate. Fructose 6-phosphate converted to fructose 1,6-bisphosphate. 1 more ATP was used. Then fructose 1,6-bisphosphate converted into glyceraldehyde and dihydroxyacetone. Glyceraldehyde and dihydroxyacetone are interconvertible. These are the isomers. Then glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate. Here 2 NADH are produced. 2 NADH are produced from 2 NAD plus. Okay. Then 3-phosphoglycerate forms 2-phosphoglycerate and 3-phosphoglycerate converts 2-phosphoglycerate. 2-phosphoglycerate converts phosphoenol pyruvate and then it converts into 2-pyruvate. Here also 2 ATPs are produced from 2 ADP. Okay. Sorry. Here 2 ATP will be produced. 2 ATPs are produced and here 2 NADH is produced. DH. So first step, this is the first step. ATP is utilized in first step and third step. Okay, ATP utilized in first and third step. Now let's see where the ATPs are produced. ATPs are produced in this is the second step, the third, fourth, this is the fifth, this is the sixth, the seventh, this is the eighth, the ninth, and it is the 10th. Now 2 ATPs are produced in the 7th and the 10th step. 7th and 10th step. 4 ATPs are produced. 4 ATP produced. 2 in each step. Okay. Now NADH is also produced. NADH is produced in the 6th step. NADH produced. 2 NADH produced in 6th step. Okay, make short notes on this, at which stage what is produced. It will be easier for you to learn. And in the 9th step, 2H2O is produced. 2H2O is released. The 9th step. This is the 9th step. Okay. And also remember, glycolysis is a oxygen independent process. It can occur both in aerobic and anaerobic conditions. This is also a very important point to remember. Glycolysis is oxygen independent. Occur in aerobic as well as it can occur in aerobic condition as well as anaerobic condition. Okay. So this was all about the glycolysis pathway. Hope you like the video. Do like, share and subscribe my channel. Thank you.